Hello everyone, welcome to another video. Today I'm talking about the CRISPR technique used by scientists to edit genomes. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. If you're like me, who's not a biologist, it's highly likely that you don't really know what that means at all. CRISPR were first discovered in archaeas and later in bacteria by a scientist at the University of Alicante in Spain. He proposed that CRISPRs serve as a part of bacterial immune system, defending against invading viruses called bacteriophages. They consist of repeating sequences of genetic codes interrupted by spacer sequences, remnants of the genetic code from past invaders. The system serves as a genetic memory that helps the cell to detect and destroy invaders when they return. Scientists around the globe have figured out a way to manipulate this natural defense system of bacteria to help us. Most ethic debate around CRISPR is centered around human genome editing, so today I will be talking about those. There are many aspects of the debate. They are safety, informed consent, justice and equality, and genome editing research involve embryos. Safety. Due to the possibility of off-target effects, researchers around the globe generally agree that until germline genome editing is deemed safe through research, it should not be used for clinical reproductive purposes. The risk is simply too high. Informed consent. Some people worry that it is impossible to obtain informed consent for germline therapy because the patients affected by the edits are usually still embryo when they were being edited. Researchers are also concerned about obtaining truly informed consent from parents because the risk of germline therapy is still unknown. Justice and equality. As with many other new technologies, the cost of such technology is very high, and the concern is that if we make CRISPR technology, genome editing, available to people, then it's going to be extremely expensive, thus only the very rich can afford it. Many people have moral and religious objections to use the human embryos for research, because once you use the embryo for research, you are basically killing the embryo, which, if we're not doing research on them, could one day be developed into a live human. So in some way, you can say that by using embryos to do research, you are killing children. While NIH will not fund gene editing for human embryos at this time, many bioethical and research groups believe that research using gene editing in embryos is important for many reasons, including to address specific scientific questions about human biology, as long as it is not used for reproductive purposes at this time. Make sure to subscribe if you like the video, and thank you so much for watching.